Hello, science fans. Today, we are going to explore a very important test in chemistry. It is called the arsenic limit test. Now, arsenic is an element just like oxygen or carbon, but some forms of arsenic can be very harmful to us, even in tiny amounts. That is why we need to make sure things like our medicines, food, and water are safe. We need to check if they have too much arsenic in them. This test helps us do exactly that. It does not tell us the exact amount, but it tells us if the amount is below a safe limit. Think of it as a safety check, a go or no-go for arsenic. It's a critical part of keeping our products safe for everyone to use. So how do we find something that's practically invisible? Science. The arsenic limit test is based on a very cool principle. It was developed by a scientist named Gutzeit, which is why we call it the Gutzeit method. The whole idea is to convert any arsenic present in a sample into a gas. Yes, a gas. This gas is called arsine gas. We then let this gas react with a special piece of paper. This reaction creates a colored stain. The intensity of that stain tells us about the amount of arsenic. A darker stain means more arsenic, while a lighter stain means less. It is a brilliant and classic piece of analytical chemistry that helps protect public health every single day. This test is what we call a limit test. It is not designed to give us a precise number, like saying there are exactly 0.005 milligrams of arsenic. Instead, it is a comparison test. We prepare two setups at the same time. One uses our test sample, the substance we are checking. The other uses a standard solution, which has a known, very small, and acceptable amount of arsenic. We run the test on both and then compare the results. If the stain from our sample is lighter than the stain from the standard, we know it is within the safe limit. It is a simple, visual way to make a very important decision about safety. The reason this test is so widely used, especially in pharmacopoeias, which are official books of medicine standards, is because of its reliability and sensitivity. It can detect very, very small quantities of arsenic. This is crucial because the safe limits for arsenic are extremely low. The clever use of chemical reactions to isolate and visualize the contaminant makes it an elegant solution to a serious problem. So by turning a potentially harmful solid into a visible stain on paper, we can make a clear judgment about the safety of a product. It is all about making the invisible visible. Bill, science is cool. Let's get into the nitty-gritty of the chemistry. Consider this. The principle of the test is based on a series of chemical reactions. The first step is to make sure any arsenic in our sample is in the right form. Arsenic can exist in different chemical states, but for this test, we need it to be in its arsenious acid form. So the first thing we do is react our sample with acids. This process, often helped by other chemicals like potassium iodide, converts all the arsenic compounds into a form that is ready for the next big step. It is like getting all your ingredients prepped before you start cooking. Every chemical has a very specific job to do in this scientific recipe. Now for the main event, making the gas. Once the arsenic is in the form of arsenious acid, we add a reducing agent. A reducing agent is a substance that donates electrons in a chemical reaction. In the Gutzeit test, we often use zinc metal. When we add zinc to the acidic solution, it reacts vigorously to produce hydrogen gas. This is a very powerful reaction. This newly born or nascent hydrogen is super reactive. It immediately seeks out the arsenious acid and reacts with it. This reaction reduces the arsenious acid, turning it into arsine gas. So the solid or dissolved arsenic is now floating around as a gas, mixed in with all that hydrogen. The chemical equation looks something like this. Arsenious acid plus hydrogen gas gives us arsine gas and water. This is the heart of the Gutzeit method. We have successfully converted the arsenic from the sample into a volatile gas. This is incredibly useful because gases are much easier to separate and detect than trace amounts of solids mixed into a complex sample. The entire process happens inside a special piece of glassware, designed to contain the reaction and direct the gas exactly where we want it to go. It is a controlled chemical transformation, all happening inside one bottle. Why is this conversion to a gas so important? Imagine trying to find a single grain of black sand on a huge, dark beach. It would be almost impossible. But what if you could magically turn that one grain of sand into a puff of colored smoke? You would spot it instantly. That is what we are doing here. We are taking the tiny hidden arsenic and converting it into a chemical signal, the arsine gas, that we can easily guide and detect. This conversion is the key that unlocks our ability to see the unseeable and ensure our products are safe from this sneaky contaminant. It is a beautiful example of using chemistry to solve a real-world problem. To perform this test, we need a special piece of equipment called the Gutzeit apparatus. It sounds fancy, but it is quite simple. It is basically a wide-mouthed glass bottle with a stopper. The bottle is where all the chemical reactions happen. It is where we mix our sample, the acids, and the zinc to generate the arsine gas. The bottle needs to be big enough to hold all the liquids and allow for the bubbling reaction to occur without spilling over. Everything must be contained, so the gas we produce does not escape into the lab. Safety first, always. The bottle is the reaction chamber, the heart of our entire setup. Now let's look at the top part. The stopper that fits into the bottle's mouth has a narrow glass tube passing through it. This tube is about 20 centimeters long and has a small hole on the side. Its job is to act like a chimney, guiding the arsine gas and hydrogen gas mixture upwards, away from the liquid. But before the gas goes up the tube, we place a piece of cotton, soaked in a lead acetate solution, inside the lower part of the tube. This cotton has a very important job. Sometimes, other gases can be produced during the reaction, like hydrogen sulfide, which could interfere with our test. The lead acetate cotton acts as a filter, trapping these interfering gases and letting only the arsine and hydrogen pass through. At the very top of this glass tube, we get to the most critical part, the detector. We place a small piece of special paper, called mercuric chloride paper, over the opening. This paper is held in place by 2 clips or another special fitting. When the arsine gas flows out of the tube, it comes into direct contact with this paper. The mercuric chloride on the paper reacts with the arsine gas. This chemical reaction produces a distinct stain. The color of the stain is usually yellow, but it can turn brownish or orange depending on the amount of arsine gas. This stain is the final, visible proof that arsenic was present in our original sample. So let's review the whole setup. We have the bottle at the bottom where the gas is made. We have the long tube to guide the gas upwards. Inside the tube, we have the lead acetate cotton to purify the gas. And at the very top, we have the mercuric chloride paper to detect the gas and create a colored stain. Every single part of the Gutzeit apparatus is designed with a specific purpose in mind. It is an elegant and effective system for isolating a specific chemical, guiding it, and making it visible to the human eye. It is a fantastic piece of chemical engineering. So, how do we run the actual test? It is a tale of 2 bottles. First, we prepare the test solution. We take a specific amount of the substance we want to check for arsenic and dissolve it in water or a suitable acid inside one of the Gutzeit bottles. Then we add the other necessary chemicals. We add stannous chloride, which helps the reaction along. We add potassium iodide, which ensures all the arsenic is in the right state to be converted. We also add a little bit of zinc. This carefully prepared mixture is now our test setup, ready to reveal if it contains any hidden arsenic. Each ingredient is added in a precise order and amount. Simultaneously, in a second identical Gutzeit apparatus, we prepare the standard solution. This is our benchmark, our control. Instead of our sample, we add a very small, precisely measured amount of a standard arsenic solution. This solution contains a known quantity of arsenic that corresponds to the official safe limit. For example, if the limit is 1 part per million, our standard solution will be made to match that concentration. We add all the same chemicals, potassium iodide, stannous chloride, and zinc, to this bottle as well. This setup will produce a stain that represents the maximum allowable amount of arsenic. Once both bottles are prepared, the reactions begin. We let both setups sit for a specific amount of time, usually around 40 minutes. During this time, the zinc reacts with the acid in both bottles to produce hydrogen gas. This hydrogen, in turn, reacts with any arsenic present to form arsine gas. The gas travels up the tubes, past the lead acetate cotton filters, and finally reaches the mercuric chloride papers at the top. As the arsine gas from each bottle hits its paper, a stain begins to form. We wait patiently, letting chemistry do its work and create the visual evidence we need. After the time is up, we carefully remove the mercuric chloride papers from both the test and the standard setups. Now comes the moment of truth, the comparison. We place the 2 papers side by side and observe the stains. The standard paper will have a distinct yellow stain. We then look at the stain on the test paper. If the stain from our sample is lighter in color than the stain from the standard, or if there is no stain at all, we can declare that the sample passes the test. It means the amount of arsenic in it is below the safe limit. If the stain is darker, the sample fails. It is a simple, visual comparison that holds the key to consumer safety. Consider it done.